neuroprotective effects of specific glutamate receptors, the metabotropic glutamate receptors, in our model of Parkinson's disease, which is the lactacystin model we're going to explain. So as you know, Parkinson's disease is a kind of neurodegenerative disorder, and you have the classic motor symptoms. And it's caused by progressive degeneration of the dopaminergic cells within the substantia nigra, and also formation of Lewy bodies. And there are lots of problems with current therapies, and so mainly they fail to slow the progression of the disease. So that's what I'm looking at to see if any of these drugs, the new compounds that are coming out that target these receptors, can slow the progression of the disease. So the basal ganglia, which are linked up to the substantia nigra where the cells die, controls movement, and it's a sort of basic diagram that what happens is in the normal brain you've got the whole system working properly. The cells here don't project up anymore with their dopamine, and so it changes the system, and it means that you get problems with movement and also further damage of the substantia nigra, which is here. No longer projecting dopamine. So the glutamate receptors that I'm looking at, the metabotropic glutamate receptors, and they're a subclass of these glutamate receptors. And glutamate is excitatory in the brain, so it sort of increases activity of neural cells.
Hang on, could you just run through that again a little bit slower?
Also, glutamate causes cells to be more active, basically. And I'm looking at a subclass of these particular glutamate receptors. And the drugs that I'm looking at reduce the activity. So what happens is they can reduce some of this excess activity that you get in the brain and might end up protecting the cells. So what happens is if you've got too much activity in the brain, in certain areas of the brain, the cells, it can kill the cells if you've got too much. You want sort of balance and this homeostasis in the brain. So by targeting a specific group of these glutamate receptors with these compounds that I'm looking at, you can reduce the excess activity. That's the idea behind it. So there's three groups, there's three classes of these mGluAs, or metabotropic glutamate receptors. And so far I've just looked at one receptor within one of the classes. This is a receptor, and what happens is your compound binds to the receptor and causes these sort of downstream effects which reduce activity. So there's been lots of new compounds that have been released recently, and I'm looking at several of these. So one particular receptor, the mGluR4 receptor, has been looked at quite a lot by other researchers, and it's been shown to reduce sort of behavioural symptoms in rats that are given Parkinson's disease, and they've also been shown to protect the remaining cells that are in the brain. But nobody's looked at any of the other subtypes, so there's 8 different receptors. They've only really looked at this one so far, and there's ongoing research in lots of different areas of sort of looking at all the different 8 receptors, but one I'm looking at, which is the mGluR8 receptor, nobody's really looked at that very much yet. So this compound, DCPG for short, targets the mGluR8 receptor. The mGluR8 receptor is present throughout the brain, but it's also in the basal ganglia. So a previous PhD student that we had in our lab, he used this drug in some rat models of Parkinson's disease, and he found, giving 3 different doses over a long period of time, that the two of these doses, so 5 and 15 milligrams per kilogram, were neuroprotective. But he didn't look to see if this drug had any effects on the behaviour of the rats, so that's what we wanted to look at, as well as sort of checking how much drug is needed to produce these neuroprotective effects as well. I'm also going to be looking at the mechanisms underlying this neuroprotection, so why is
Can I interrupt
neuroprotection.
a second? Could you, I must have missed it. Where are these receptors? Are they on the ends of nerve cells, or are they at the end
Yeah,
of the
they're
axon?
found presynaptically. These
So they
specific
just have the
ones.
synapse.
Yeah.
Sorry.
When you say the various molecules have been released, who released them? Do they suitable from a health point of view or a safety point of view? Or?
Well, that's what's still being tested.
Size of the molecule?
So they haven't been tested in clinics
Oh, I see.
yet.
Okay.
They're preclinical. So we've used several different models of neurodegeneration for creating a Parkinson's model. The most common one you might have heard of is the 6-hydroxydopamine model of Parkinson's, and that causes this rapid neuronal degeneration. And I use this to test my lesion accuracy. So in the rat, we needle it directly into the substantia nigra and inject the 6-hydroxydopamine toxin, and that specifically kills the dopaminergic cells in that area. So it's recreating sort of model of Parkinson's disease. And so I used this just to test my accuracy of getting the needle in the right place in the brain, and that went fine. So then I went on to do exactly the same thing but with lactacystin, which is a proteasome inhibitor. So basically it stops proteins from being broken down, and you get an accumulation of proteins, unwanted proteins basically, and this causes the cells to gradually die, and it's a slightly slower model of neurodegeneration than this model of Parkinson's. So the idea is that if I can start treating the animals with the drug earlier on, sort of after the lesion, but early on in the lesion progression, then it's the equivalent to maybe an earlier stage of Parkinson's disease. Whereas most people that do these drug studies, they use maybe a 6-hydroxydopamine model, and they give the drug before lesioning and also straight after, and that's not very clinically relevant really, because you obviously can't do that in people. You have to give the drug after the symptoms have started, so after the neurodegeneration has started. And I've also recently been looking at another model called LPS model, where you inject lipopolysaccharide, which is taken from bacteria, and that causes this immune response in that area of the brain and rapid cell death.
Sorry, what was that last one? What was it? The last LPS?
It's called lipopolysaccharide. And basically it allows you to see if the drug that I'm giving the rats can affect the inflammatory response, because inflammation is associated with the degeneration of the brain. So I've been looking at that as well. So these are my main aims. I've got to look at different mGluR ligands, so different drugs that target these receptors, and I'm studying the neuroprotective and the behavioural effects of these on the lactacystin model, particularly on Parkinson's disease. And I'm also going to look at their mechanisms of action underlying this neuroprotection and behavioural effect, and also their potential to be used as candidate drugs, maybe in clinical trials later on, and prevent sort of further neuronal loss. So my basic study design is this. So I take behavioural recordings before I've lesioned the animals, and then I'll lesion them, and that counts as day 0. And then 4 days later, I'll start dosing with the drug of interest for 14 days, for 2 weeks. And so after 7 days since lesioning, I'll do another behavioural test. 14 days later, I'll do another behavioural test. And at day 18 is the final day, so I've been dosing with the drug for 14 days by that stage. And I do the same behavioural test again, which I'll run through in a second. And also these amphetamine rotation tests right to the end, which I'll go to explain in a second. So, as I said, I do a stereotactic surgery, which is basically putting the needle directly into the substantia nigra. And I either give lactacystin or this lipopolysaccharide. And then I start dosing from day 4 with either saline, so that's like a control group, or a low dose and a high dose of this drug, basically. And behavioural testing involves doing a vertical cylinder test, which measures the use of the full force. So when we lesion the rat brain, we just lesion one hemisphere, which means the other hemisphere is there as a control, and any motor deficits that the rat develops will just be on one side of the body. So we lesion the left it means that it affects the right side of the body. And you can pick up this asymmetry with the behavioural tests. And then right at the end of the study is this amphetamine rotation test. I'll show you the little video of the vertical cylinder test, and we measure the use of the forepaws. So rats naturally rear in a new environment, and because they're in this narrow cylinder, they'll naturally rear up. And we can measure the use of which forepaw they explore with. And then with the amphetamine rotations, what we do is when you give the rat some amphetamine at the end of the study, it tells you the extent of the lesion, so how big the lesion is. And if the more they spin, the larger the lesion. So the idea is if the drug is neuroprotective, they won't spin as much because the lesion won't be as large. So my methods, just remove the brains, and then I cut thin sections in a freezing cryostat. Just takes really thin sections, 20 microns thick. And then I do some immunostaining. So I don't know if you've heard of this technique before, but you use antibodies to specifically label different cells within the brain. So I can stain for either dopaminergic cells, brown stained cells, and I can also stain for just all the other types of neurons in that area. So it just stains purple. And I can also use this antibody OX6 to target specifically activated microglia, which are involved in the immune response in the brain. So if there's lots of these cells, it suggests that there's an increase in the immune activation in the brain. So these are my results so far. So the lactacystin lesion DCPG treated rats. The behavioural tests show that, and this is quite positive, we've got in the vertical cylinder test, which I showed you previously, in the control group, which is the red, fully treated, you get a gradual decline over the 14 days of lesioning. You get a gradual decline in their use of their forepaw, so it shows that the lesion is getting bigger and that they're unable to use their forepaw as much. Although in the drug-treated animals, so both the low dose and the high dose, you can see that actually they're significantly different from this control group, but actually they stay the same as baseline. So the drug is having effects on the behaviour, and it seems to reverse the behavioural deficits that the rats would normally show. Although in my amphetamine rotation data, and this is all preliminary results, so I'm still analysing some of the results that I've got, you don't seem to see much change. So I'm hoping that that will change as I sort of finalise my data. With the immunostaining and quantification, so what we do is we stain the antibodies, and then I can count them using special equipment on the computer. And this allows you to count each neuron in a specific area and estimate the number of cells that are left in each hemisphere, in each substantial micro. And you can see that these Th-positive neurons, which are dopaminergic cells, you get a gradual, a stepwise sort of decrease depending on the dose given. So in the control group, you nearly use all of the dopaminergic cells, whereas there seems to be some neuroprotection in especially in the high dose group, but also in the low dose group drug. So there is some neuroprotection going on, but again, this is still preliminary results, so I'm hoping that I get some significance and that it's actually quite a clear difference. And then this neurons should show a similar pattern. I've also looked at these activated microglia, the immune cells of the brain, and you can see the substantial microglia, it's like in a triangle here, and you can see all the activated microglia kind of flocked to the dopaminergic region, and the more there are, so basically in the drug-treated animals, there's a reduction in this inflammatory response. So thinking about the drug's efficacy, so maybe the mechanisms that are underlying it, it might be that it just modulates the transmission in the brain, much like might reduce the excitotoxic damage that happens by reducing all the excess lactic activity. It might reduce this innate inflammatory response by reducing the number of activated microglia. It might protect the neurons from cell death in that way. Also, Hugh, who did his PhD before me and was looking at a similar thing, he found there was a reduction in these alpha-synuclein deposits linked to Lewy bodies. And so I'm going to have a look at that as well. I haven't had a chance to do that yet. So from my data so far, I can say that DCPG does reverse these behavioural deficits, and this might be due to the modification of signalling within the basal ganglia. It may be neuroprotective, although this is yet to be confirmed at both the doses given. So it's good that it's still equally neuroprotective at the low dose. DCPG does reduce the number of activated microglia in the substantia nigra, and so it might be having an effect on the innate inflammatory response and dampening this down. And also, to conclude, targeting this specific receptor might be a novel therapeutic strategy for treatment in early stage Parkinsons, by slowing this progressive neurodegeneration. So my plan is next to finish off the studies that I've been looking at, and hopefully with the preliminary results just finalise the data. I've got to finish off the LPS lesions, the lipid polysaccharide lesioned animals, because this will confirm that the drug has an effect on the inflammatory response, because LPS causes an inflammatory response, so if the drug dampens this down, then it's definitely having an effect in that area. I'm also going to do a few other investigations with this drug, so seeing if an acute drug treatment, so just giving the animal one dose of the drug, if that has an effect on its behaviour, and also withdrawal, so seeing if taking it once you've dosed the drug for 14 days, seeing if the animal returns to its previous lesion state. So basically, that would suggest that the drug's having an effect constantly rather than just being neuroprotective and protecting against the lesion. I'm also going to look at a few more behavioural tests to see if I can notice any small changes. And I'm going to be looking at some other new compounds that are coming out. So I've done this short study with this drug, which is an mGluR7 agonist, which so the previous drug was an mGluR8, so I'm just looking at a few different receptors. And then at the end of my study, I'm going to look at cell culture to have a look at some of the underlying mechanisms of neuroprotection and try and work out what's happening in the brain when I give these drugs. So I'd like to thank David and Ian, who I work with, who's the fellow PhD student in my lab, and the whole of the Dexter lab group, as well as Parkinsons UK for funding and research.
Thank you.
Thank you.
Any questions?
Yes.
How many rats did you try this one to get the results?
So far, I think it's about 10 or 11 in each group, and I'm still processing them. So I've got a sort of end of. So for some of the data, I've got. Okay. So the higher the number, the smaller the error bars, and kind of the more realistic the data will be. But obviously, we don't want too high an n number, so because we want to minimise the number of animals we use. So I've got an n of 8 in the same group and 10 or 11 in the other two groups. That's because some of the animals either weren't sort of very well lesioned, or they didn't sort of survive very well after the surgery. That doesn't happen very often. But
Is that statistically insignificant?
yeah, so this behavioural test is, so the stars give me different significance. That's pretty significant.
You're not.
Could I ask some questions, please, about the effects of the surgically reduced lesioning?
Mm-hmm.
This is assumed to be a model, produce a model of an analogue of Parkinson's symptomology in the rat.
Yeah.
Can you tell us what that symptomology is and how closely it resembles that in human Parkinson's? I mean, is there an asymmetric or symmetric tremor? Is there slowness of coordination? Are there other symptoms which are not observed in humans with Parkinson's?
I think it's usually the other way around. So there's symptoms that humans get that you obviously it's hard to measure in rats. So we stick to mainly the motor symptoms, and you get a lot of non-motor symptoms in human Parkinson's disease, obviously. But basically, this lactacystin model, we use it because it's a slightly slower lesion. So if you use 6-hydroxydopamine, almost all of the cells are gone by the next day, 2 days later. So using this model, it sort of allows a slower degeneration, which means if you can kind of get in that window where it's just beginning to degenerate, with the drug, you can see if it would be neuroprotective or not. Whereas if all the cells are already dead and you're then giving the drug, you're not going to see any effect. You won't know if it's neuroprotective or not. But yeah, there are always issues with these animal models. They're never identical to the human equivalent. But they're sort of gradually trying to get better models as we go along. But this is the one that we've chosen to use.
Can I ask a very basic question about where the lesions actually occur? Is it in a corresponding part of the rat's brain to the strata nigra?
Yes.
Is that where
Yes,
it
that's
is?
exactly right.
Okay.
Yeah.
And then immediately after the lesions are induced, what behavioural changes are observed?
Immediately after or gradually?
Well, okay, not immediately, then in the
Gradually
next
over time. They
observation.
gradually use the ball on the opposite side of the body, so the hemisphere that's controlling the other side of the
Yes.
body, they gradually use it less and less and it becomes weaker, so they tend to kind of hold it in.
Is there a tremor?
No.
You won't see that in an animal model. You might see it in a monkey, but not in a rat. You can't see tremor in rats.
And is there an explanation?
No. I think it's just because of the animal. With being so small, I think in a primate, also the neuronal networks are much more similar to what they are in humans. And I think it's just the size of the animal, I think, as well. You don't see a tremor. And it's more that generally they're actually resting on their paws all the time, so you don't see so much of a tremor. You know, like you would have somebody with their hands, relaxed in their lap or something like that, you might see a tremor. Generally, the animal is actually walking on the paw, so you might not see the tremor.
So the model rests upon the assumption that biochemistry is the same.
Yes, but also the motor pathways aren't the same, so this is where you're seeing the deficits. So when you put the animal in the cylinder, what it's doing is exploring to try and find a way out, and it's using both its left and its right hand paws. But as the lesion grows, it will tend to be using the left hand paw more to move around, and you'll be tending to use less the right hand paw. So you've got a motor deficit on one side of the body. So you can see this change gradually progressing.
But am I right in thinking that such an asymmetrical limb deficit, movement deficit, could be induced by lesions in a different location?
Yes.
I mean neighbouring.
Yes, you could lesion them in all different areas of the basal ganglia, but what we're trying to do is trying to mimic the principal cell loss within the nigrostriatal pathway that you see in Parkinson's. Yes. And again, the circling behaviour, amphetamine, like if you give it amphetamine to a human, it would make them hyperactive. And if you give it to a rat, it becomes hyperactive and it would be running all over the place. But because you've lesioned one side of the brain, what it tends to do is circle rather than walking it in a straight line. So again, these circling behaviours, are characteristic of the size of the lesion, the number of turns.
To put it crudely, if you looked at an animal with this kind of lesion you knew the symptoms of human Parkinson's, unless you knew the location of the lesion, you wouldn't be able to categorise it as suffering from Parkinson's.
But we know where the lesion is.
Yes,
Yes.
but you admitted that such a behavioural disturbance could be induced by a lesion in another part of the brain.
But we verified where the lesion is.
Yes, I see that.
Yes.
It would
What-
have to be linked to the motor
What-
neuron.
Yes.
I mean, the ideal case would be to have an animal model that displays all or most of the human symptomology of Parkinson's, which emerges from a lesion in the striata nigra and doesn't if you place the lesion elsewhere. Is that right?
Well, the thing is, if you chop somebody's spinal cord
Yeah,
on one
yeah,
side,
right.
it will affect one side of the
Sure.
body. So that is a very severe example of it. But what we're trying to do is mimic the pathway degeneration. So we're targeting it very specifically with this toxin. You know, if we put it in the cortex, you wouldn't see these behavioural changes. You know, if we put it in the cerebellum, you wouldn't see these behavioural changes. So it's very specific to this area.
Thank you.
And there is no perfect model of Parkinson's
No.
disease. We've been
That's
trying for, 60 years to try and get one, and we still haven't got a perfect model. Yeah, and even the genetic models are far